the Slack. I bet there's going to be more questions for you here. We'll go ahead and switch over to our last speaker of the day, Dr. Kelly Jen, a recent PhD graduate from the M3D program in the Department of Lab Medicine and Pathology. And, you know, I don't have your official title now that you're over at the Allen Institute. What do they call you over there? Oh, it looks like you're muted. You can always like mime it out too.
Oh, sorry. I'm a scientist.
Scientist. All right, cool. She continues to be a scientist and an excellent one. So she's going to be telling us about the canine epigenetic clock. And we'll go ahead and have you take it away, Kelly.
Thanks, Mitchell. Is the screen okay?
Looks awesome.
So thank you again, Mitchell and Alessandro, for inviting me to speak. And I'm really excited to talk to you about a project that I started as a PhD student and that's still ongoing, called the Canine Epigenetic Clock. So as I'm sure all of us know, aging varies widely across the animal kingdom, as we can see by the species shown on the screen and the enormous variation in maximum lifespan across all these species. But as we all probably also know, aging varies quite widely within single species, and this is perhaps demonstrated no better than through the most phenotypically diverse mammal that we know of, and that is the companion dog. So Canis lupus familiaris, or the domestic dog, is of course incredibly morphologically diverse, but it's also very diverse in terms of how different individual dogs age. So some of us probably know that larger breeds tend to live shorter than smaller breeds. For example, Lou, the Irish Wolfhound here, tends to have a mean lifespan of about 9 years compared to Chihuahuas that live much, much longer. And here are just these two same animals shown at 9 years of age. Right away, we can see that there's a big difference in the young and the old picture of Lou. Probably the most striking difference being the change in coat color, but of course there's lots of changes that the camera doesn't capture, including changes in behavior, as well as different diseases Lou might have acquired in his lifetime. On the other hand, Toto, to me at least, doesn't look too different between 9 and 3 years of age. And this, again, just kind of demonstrates, even though these two photos are showing these animals at the same chronological age, 9 years, they're both capturing very different life stages in these animals' lives. So now an interesting question becomes what causes this variation in aging? And I don't think anyone is surprised to hear that it's some sort of mixture between genes and environment. And this leads to kind of the big picture that a lot of us in the Promise Lab are interested in, which is how can we use naturally occurring genetic variation to learn more about complex phenotypes like aging. So in addition to what I've already said, there's lots of great reasons to use dogs to study aging and disease. Centuries of artificial selection have resulted in this really unique population structure that we know of as breeds. High levels of genetic homogeneity within breeds are coupled with just a lot of heterogeneity or diversity between breeds, which we can use to our advantage. Furthermore, dogs and humans also die of very similar kinds of diseases, perhaps with the exception of cardiovascular disease, which for some reason dogs don't really die of very often. And of course, dogs share our environment in a way that really can't ever be replicated in a lab. So this lets us ask important questions about key environmental factors that influence all different kinds of disease, things like smoking status, air quality, and socioeconomic status, just to name a few. And of course, the amount and the quality of data that are out there for dogs is really second only to people. So there's just a ton of information out there waiting to be mined by scientists. So to try and get at this question of how genes and environment influence aging, we thought that a good place to start would be the epigenome, particularly this kind of recent phenomenon that a lot of people have been studying called the epigenetic clock. And that's this idea that many scientists now have found that just by simply measuring a handful of DNA methylation sites, we can extremely accurately predict chronological age in individuals. And shown on the screen are two figures taken probably from the most widely cited of these studies from Steve Horvath, showing these clocks or these predictors that he's built from different human tissues. For both of these plots, predicted age is shown on the y-axis, and the actual age of the people are shown on the x-axis of the observed age. And these are two predictors he's built, one using white blood cells and one using breast tissue, and we can see that there's tight correlations between the predicted and the observed ages. But perhaps what's more exciting about these clocks, besides their ability to predict chronological age, is perhaps what they may say about biological age or the overall health of an organism. And so this is a cartoon taken from a review kind of demonstrating that idea, where these three individuals all happen to be the same age, but their predicted ages are different. And some people have argued that their predicted ages correlate with their overall health. So given all of this, we were really curious to ask what we can learn about epigenetics and aging from this exciting new model system. So of course, to do this, first we needed a set of a sample population of dogs. So with help from our collaborators over at Texas A&M Veterinary School, we got a cohort of 70 healthy dogs, and from their blood samples, we measured two different kinds of epigenetic profiles. The first was DNA methylation, as measured by reduced representation by sulfite sequencing, and also chromatin accessibility, as measured by a TAC-seq. And unfortunately, I don't have time to get into the details of the methods, but to kind of cut to the chase, the final data sets that we had to work with looked something like this. So for both kinds of data, we had about 15,000 features each for all 70 dogs. And again, this is just an enormous data set, which allows us to ask lots of different kinds of questions. But today I'm going to talk about just a few. And the first one being kind of the most basic question, which is, can we even build a predictive clock that predicts age for dogs using either of these two data sets? And to do this, I used a machine learning method called elastic net regression. And that's the same method that Steve Horvath and Greg Hannum had used to build their clocks. And so I'm going to show you the results on the next slide. So again, I'm showing you predicted versus observed age from the elastic net model that I produced using only DNA methylation data. And again, this is only 70 dogs. So we can see right away that this correlation is pretty tight, an R-squared value of 92%. 100% is a perfect predictor. So this, indeed, this DNA methylation clock is, I think, performing pretty well. If we do the same thing with the TAC-seq data, we also see a pretty tight correlation between predicted and observed age, although performing not quite as well as the DNA methylation data, but still pretty well at 80%. And so to me, I concluded that both of these data sets independently can predict age pretty well. So the next question, now that we have these clocks, these predictors, I wanted to ask if and what they may tell us about biological age. And to look at that, to ask that, I looked at something called age acceleration, which is a pretty simple calculation. It's just the difference between predicted and observed age. And to kind of conceptually kind of demonstrate what that means, if we look at our cartoon and we want to measure the age acceleration values of these two individuals, this individual on the top, who's perhaps more frail, or some may say aging faster than his age-matched peers, would have a positive age acceleration value since he's above the isometric line. And on the other hand, this fellow down here, which is who seems to be more sprightly compared to his peers, would have a negative age acceleration value. And so if we apply this to to our dogs, kind of a straightforward hypothesis emerges, since we know that our large breeds tend to be shorter lived than our small breeds. And if I were to kind of predict where I think they would fall on this plot, I would think these larger breeds would have larger age acceleration values than these smaller breeds. And kind of plotted a different way, if we just directly plot age acceleration over breed size, if our hypothesis is true, then I would expect to see a more positive correlation. So what did we see? So it turns out there is no correlation between age acceleration and breed size. So far there's no evidence supporting this idea that larger breeds are aging faster, at least based on the clocks that I built here. Okay, so moving on to another question, a third question. I finally wanted to kind of compare these two different clocks and ask if they are in agreement in terms of what they might be telling us about the biology behind aging. And so to do this, I looked at a couple of different things that I'll talk about today. The first thing I wanted to ask was if there was any genetic overlap between the sites selected for these two clocks. So the model building algorithm selects sites, features from each of these models that it thinks are most important. So I was wondering if it would select, if these important sites would be coming from similar regions of the genome or similar genes. And so to cut to the chase, no, there was no overlap between these sites that these two clocks were calling important. So then I moved on to kind of another strategy, and that's looking at direct age-associated sites. And I'll talk a little bit more about what I mean by that. So everything I've shown you so far has been the result of this machine learning method called elastic net modeling, which uses multiple features to predict age. I took a step back and did something a little bit more basic. So going back to just basic linear modeling, which allows us to assess individual features one at a time and find their relationship with different predictors, including age. So just as kind of a visual representation of what this means, I would model each individual epigenetic feature, so each column in these data sets, as a function of 3 different predictors: age, weight, and sex. I'm just going to talk about age for today. But what this gets me is for each feature, for each individual column in these data sets, I'd get a single p-value estimate for each predictor. So these are the results. These are the number of features associated with each predictor. And so what this number explicitly means, 110, is that out of the 14,000 or so methylation sites that we measured, 110 of them were significantly associated with age. In contrast, over 1,000 out of the 15,000 ATAC-seq sites were significantly associated with age. So this is interesting in itself, since if you remember earlier, our methylation clock was a little bit better at predicting age than our ATAC-seq clock, and yet our ATAC-seq clock has our ATAC-seq measures many more features that are actually significantly associated with age. So next, I wanted to ask if there were any interesting ontology terms that came up when I looked at these individual features and the genes that they fell near. So after performing enrichment analysis, these were the results. So for these 110 age-associated DNA methylation features, the genes that they happen to be near seem to have to do with these 2 different developmental terms, or embryonic organ morphogenesis and head development. Interestingly, a very different set of terms came up for the age-associated ATAC features. And I'm not going to read all of them, but kind of what stood out to me was a bunch of terms having to do with mitochondrial function and structure, which again is interesting because there's a lot of ongoing research kind of trying to tease apart the relationship between mitochondrial metabolism and epigenetic changes in epigenetic structure. So I think that was kind of an interesting thing to see. And so I wanted to dig even deeper into the the relationship between these features to see how they compare to one another. So what I did was I looked at the correlation structure between each of these sets of features. So for these 110 DNA methylation features that were associated with age, I correlated them all with each other to kind of see what they look like. So what I'm going to show you next is 110 by 110 matrix of correlation coefficients. And they're colored by the strength of the correlation. And so we see here, it looks like there's some structure there. And if we look at the distribution of these coefficients, they center around 0. And now when I show you the same thing for the attack data, there's kind of a pretty striking difference. So there's obviously much more structure within these age-associated attack-seek features. And if we look at the distribution, they don't center around 0. Instead, they center around an absolute value of about 0.4 in terms of correlation. And all of this is just to kind of highlight the differences between how these features relate to one another and how they relate to age. And so just, I guess, the take-home message I kind of wanted to say, given all of these things that I've shown you, is that it kind of appears to me that even though both of these data sets are able to pretty accurately predict age, it seems like they may be telling us different things in terms of the biology. And so just to summarize everything I've talked about, we were able to successfully build these 2 different clocks to predict age in dogs. And this is the first time that I know of that anyone's built a clock using attack-seek data, so that is pretty exciting. And despite the fact that both of these clocks can predict age, they seem to kind of tell us different things in terms of the biology that might underlie these different correlations. And so it seems like, you know, most, if not all of the field is really focused on DNA methylation right now in terms of predicting age, but it seems like there's a lot of stuff we can learn if we look at other parts of the epigenome too. And of course, there's a lot of caveats to keep in mind. This was a really, really small sample size, but at least that one particular caveat can be addressed by future efforts that are currently underway through the Dog Aging Project. And hopefully I've convinced you through this short story that we can use, you know, creative model species like the dog to study naturally occurring genetic variation that can teach us about aging. So with that, I want to thank the lab and all of our collaborators. This was an enormous group effort from a lot of people. And I am officially at the Allen Institute now, but I'm still affiliated with the lab and I'm still actively working on this project. So feel free to contact me at either of those email addresses on the screen. And thank you for listening, and I'm happy to take questions.
Awesome. Great talk, Kelly. So Niran Haddad has a question for you. So are the prediction values based on all data, test data, or leave one out?
Leave one out.
Leave one out. Okay. And I guess you both know what that means, so we'll just go on. Dr. Bito has a question for you. I'll let him go ahead and ask it.
Oh yeah, no, very quickly, Kelly, that's fascinating talk. I was wondering, so I think the major determinant for the correlation between dog breed size and longevity has been determined to be circulating levels of insulin-like growth factor 1, IGF-1. So have you tried, instead of plotting against breed size, looking at, if you have those data, looking at serum level of IGF-1, circulating levels of IGF-1 in those dogs?
Yeah, that's a good idea. We don't have that information, and I'd imagine it'd be kind of hard to, even if we still had access to these exact same dogs, I would imagine those levels change a lot based on circadian patterns and things like that. So it seems really require a pretty coordinated effort from our vets to do that.
Yeah, probably yes.
Well, you know, Peter, he's actually got a question that might allow you to kind of map that onto there a little bit. So he was curious, are there subsets of loci in either of the clocks that do have the expected correlations with breed size? And then if you think about IGF-1 and breed size having a relationship to each other, you might kind of be able to bootstrap yourself to that.
Yeah, I haven't done that directly. I don't think, looking at sites that correlate directly with breed size. And I am a little bit hesitant to rely too heavily on those breed sizes. I didn't mention in the talk, but those are kind of estimates They weren't like the actual weight of the actual individual animals. They were based on kind of owner reports of the breed, and then we looked up kind of a general, like standardized breed weight. So there's always a little bit of uncertainty there, but that's also an interesting idea.
Very cool. Just one more brief one. Yeah, given that you found so much difference between the two clocks, and I know you're making the point that we can learn different things based off of each one. But if we had just one clock to construct, which would you be more comfortable thinking about in terms of, I don't know, maybe the interestingness of the information you get out of it, or the veracity of the insights? How would you engage that?
I mean, there's certainly a lot more information, a lot more people who've invested effort into the methylation clocks, and they've certainly been, tried and true. A lot of people have, used Horvath's algorithm in particular and shown that you can very accurately predict age. But as for what they tell us about the biology and biological age, I think that's still a really open question. I wouldn't want to endorse any clock yet in terms of what they are telling us about the biology of aging and biological age. But it's certainly, I think it's really interesting to explore different clocks and kind of not limit ourselves to a single one. And it's hard to say, but it's a really active area of research, and I think it's really awesome how different people are now making clocks in different organisms and seeing different kinds of patterns. So
that's very cool. Yeah, very fascinating. You do have some other questions in the Slack. I'll let you go ahead and have a look at those. But yeah, we're just about at the end of our day today. So just thanks are in order.